Thank you. I have been mentioned quite a bit. It's kind of fun to see that EXO at least got some other efforts started. So I'm going to talk about the comparative toxicogenomics database just as a model for, I guess I'm sort of preaching to the choir. We know how to reuse data and integrate existing standards. But I'm going to focus on two of the sort of newer initiatives that we've been adding to this database in recent years. So for those of you that may not be familiar with CTD, it essentially, the goal is to try to help investigators identify connections between chemical exposures and disease or phenotypic outcomes now via identifying what those underlying mechanisms might be. And so in the early days, it's now been like a long time. I realize how old I'm getting because this project has been around a while. It was originally just to sort of connect chemicals with genes and proteins and make those connections to diseases. So all of the data that we bring to the table is manually curated from the literature, but we also incorporate a number of other public data sets to kind of flesh out some of the context for the project. So let's see. Okay, so everybody that was talking before me kind of laid the foundation, so I'm not going to spend time justifying why we need standards and ontologies. But basically, we use those at CTD in the same way that everybody else does to sort of help with interpretation of what is otherwise sort of disparate data in the public domain, to help with extrapolation across different kinds of systems, whether it's in vitro, animal model data, or human data. Certainly for curation, which is a large focus of our project because we're tasked with going into the literature and finding information and capturing it in a very systematic way so that we all know what we're talking about and you as a user of our resource know what we're talking about. And then of course to integrate external data sets. So as other folks mentioned, genes and proteins are sort of no longer an issue, but some of these other contexts are a little bit more complicated to capture. And while there are a lot of resources out there, we're definitely not all using the same ones. And particularly for chemicals, I think we've heard 3 or 4 different resources, and we use yet another one that has not been mentioned. And phenotypes kind of stole this from Melissa Handel, which I think is a really great description. And Chris sort of laid the foundation for the complexity of capturing phenotypes. And we have kind of a different approach for doing it, but we've all talked over the years about how eventually we might be able to converge on a similar approach. So this is where we are. Chemicals, we use mesh as a structure. It's not ideal, but the reason we don't use KEBI, for instance, which a lot of people use, is only about 60% of the chemical space that we have curated data for is represented in KEBI. And so if we were to merge over to it, we would lose a lot of our data. So in the absence of having KEBI sort of pull in those additional information, we're kind of stuck with mesh at the moment. Genes and diseases, we use EntreGene. We were originally using just mesh for diseases, but a couple of years ago, many years ago now, we took OMIM and kind of mapped it to mesh and called it MEDIC. And then we also have kind of a MEDIC SLIM that allows us to sort of categorize big groups of diseases. Exposure, EXO was mentioned a few times. So that was an initiative that took many years, despite the sort of small infrastructure that it was meant to model. So I'll talk about the details of that. So exposure and phenotypes are really what I'm going to talk about in terms of the challenges we've had to face with that. So EXO was as I mentioned, talked about a few times, but I'll just take you back in time a little bit. We were approached because at the time, CTD was really one of the only environmental chemical focused databases out there, and the exposure community got in touch with us and said, hey, can you incorporate this information? Had no idea what the exposure community really wanted. So we spent a lot of time with a core group, including Elaine in the back, and said basically what is exposure data and what are all the parameters and how do we capture this? And then we went out and looked for ontologies and couldn't really find something that collectively represented all of that. So a couple of years later, we ended up with what we called EXO, and essentially the high-level characteristics of this was a stressor. And in our world, the stressors are chemicals, but we developed this framework so that others could kind of plug into it. So a stressor could be, psychosocial stress. It could be all kinds of things, but for the purposes of our database, we were focusing on chemicals. Receptors are, the population essentially of who or what is being exposed. The exposure event is all of those parameters, like what is the route of exposure, what was the timing of exposure, things like that. And outcome, the focus for us was diseases. So the way we developed this, we are not ontologists, and unlike some of the earlier speakers, I don't love ontologies. I find them like really, really hard to develop. But so what we did was kind of take a different approach in the sense that once we had that sort of overarching framework, our curators started curating the data, and we started figuring out, well, what kind of information is actually in the literature, and we'll build out the terms and try to keep them consistent with each other for a certain period of time, which we did for several years, and then started looking out to other ontologies, because obviously people have talked about unit ontologies and assay ontologies, things like that, that we could plug in and say, oh, okay, we're using all these terms, but they actually exist in other ontologies, so mapping those over. So we really, one of my curators likes to refer to it as foliating. So that's kind of what we did. We had this framework and then just started curating, and it has worked well for us. So I'll show you kind of what that looked like. So we have these stressors, we have source information and chemicals, we have receptor information. So a lot of these kinds of concepts that were talked about for the cheer resource as well. For exposure events, and this is not a comprehensive list. These are just kind of meant to show you the scope of data that we were sort of charged with capturing. So what was actually measured, and what was the medium in which it was measured? Is it blood? Is it urine? Is it air? And then timing of the exposure event, and then outcomes, which were initially diseases, but now that we're capturing phenotypes, we have that information as well. So we capture this information, and this is not a complete set of the characteristics, but you can see there are about 35 different fields of data that we capture for exposure studies, which is really different from the rest of our data, which was very almost binary, like a chemical interacts with a gene, and there are some qualifications in there, like it increases mRNA expression or protein activity, things like that. So this is really time consuming, but I hope that I can show you that there's some value in getting this information in the format that we've come up with. It would be wonderful, and we have a paper where we kind of ripped on the epi community a little, in that it was like, can you guys all get together and start talking about your data in a similar way? Because then the kinds of statistical models that are used, the kind of units that people use, it's really, really complicated to try to get it in a standardized way. So this was not fun to figure out. So the implications for our users, being able to see this information, you probably can't read it so well, but essentially in CTD, you can come at the data in any which way you want, from a gene perspective, from a chemical perspective, from a disease. And this is just an example of cadmium. And we have these tabs across the top. If you're looking at a chemical page, for those of you that might not be familiar with it, we now have these exposure tabs. And essentially what this allows you to do is say, for your chemical of interest in this example, where are all the studies that are epidemiological or exposure studies that deal with cadmium? We give you information about the references always provided, what the stressor was, what was measured, information about the population that was exposed. And then in some cases, oh, I forgot I had this. So yeah, all this different information. And then if you want the actual measurements of how much cadmium was measured in a particular population, not all studies report that, but where they do, we include that information as well. We've added some capabilities in the last year or so. So all of these characteristics up here represent the information that we capture for these particular studies. And if by default we showed you all of that, the page would be enormous and you'd be scrolling over to the side for a long time. So what we do is sort of have a default page, but there's a ton of information that you can get. And so you can kind of click and unclick what you want to see and download more or less customizable views of the data. Okay. So one of the examples we tried to, once we launched these data, we wanted to say, you know, what is this going to really do? Because part of this was to centralize this kind of information, which previously was not captured in a public database. And this is a heat map that what we did as a pilot study was took all the ag health study papers, and there were, at the time of this publication, about 110 of them, and nobody had looked at, you know, what are all the findings that people have reported for this really important epidemiological cohort. And what we were able to do with the data was to say, essentially, here are your diseases and phenotypes. These are all the chemicals that have been measured in this study. Different studies focus on different aspects of the cohort, and then look at, well, what kinds of diseases are reported as having positive associations with exposures to different chemicals. And you can start to see correlations that you might not have been able to see otherwise. So this collection of chemicals are represented in these columns, and then they correspond to different diseases that you might not really associate with one another. So by virtue of, in some ways, the exposure data is a standalone, but because we've incorporated it into CTD, we also have all of the other mechanistic data for these chemicals in both human and model organism studies. So what you can do then is say, okay, well, this chemical was associated with prostate cancer in this epidemiological study. This chemical and prostate cancer are associated with this collection of genes. And so you can start to make connections then and say, okay, well, what does it look like for diabetes? Well, in prostate cancer, across this collection of chemicals that we saw on the previous page, there's a common set of 29 genes. And between diabetes and prostate cancer, there's a common list of genes. So I hope that made sense. So you're not just looking at the exposure data then, you're able to look at it in a mechanistic context. And in most of those epidemiological studies, there's no mechanistic data, right? That's not the charge of that study. It's really just to make a connection between the chemical exposure and some kind of outcome. But if you can bring in all those sort of experimental data sets with it, you can start to fill in some of those blanks. So, and we did a recent review of cadmium and looked at all the epidemiological data we had for it, brought in all the model system data we had for cadmium, with a particular focus on metabolic disorders. And this paper was just came out a few months ago, trying to show that this integration of this human population-based data with experimental data is actually a useful approach. Okay, so then the phenotype module. So we describe phenotypes as biological outcomes that are not inherently a disease, right? So we had this sort of chemical gene disease thing, but there's a lot of stuff that goes on in between that, right, before you actually get to the disease. And we felt that that wasn't captured adequately in our database. And we looked around at phenotype stuff. There's a lot of phenotype ontologies for different systems, and Chris and Melissa have done a lot of work, some of which you heard about with phenotypes. And we were trying to figure out, well, how can we leverage some of those same approaches? We had done a pilot study in a collaboration we had with some Pfizer investigators early on, where we used mesh. So we had some preliminary data that we were trying to sort of fit into existing approaches. What we ended up doing, which is not necessarily ideal, but worked for us because of the ontologies and vocabularies that we were already working with, was to use the gene ontology. So examples of like a phenotype versus a full-fledged disease are here. So abnormal cell proliferation in the lung could be associated with lung cancer. So just trying to explain that difference. So our phenotype curation is pretty complex. So we have a chemical, we have some sort of qualifier. So the effect is increased or decreased. We have an entity. This is what we use the gene ontology for. We have anatomical terms that we add to it, right? Because we want to know where in the body something is occurring. The particular species, whether it's an in vivo or in vitro. And then a lot of people we've started capturing whether the information came just from the abstract or full text, because sometimes people want to know that. And then finally, the source. So what this looks like now is that you can actually search by phenotypes. And similar to the exposure data is, not only is the phenotype going to be associated with the disease, but you can get mechanistic data associated with those phenotypes. So if you were to look up something like leukocyte migration, you would come to a page in CTD where you could connect that phenotype with particular chemicals as well as with genes, because all that information is already there. We also have tools where you can build some pathways in there. And then, so the other value added, I think, to this is normally we would have had, just as an example, cadmium via some genes, we can make an inference to a disease. So cadmium affects this set of genes. We know that this set of genes is associated with Alzheimer's disease. Now we can actually use our phenotype information to do something similar. So we might have cadmium is associated with Alzheimer's disease over here. Cadmium is also associated with these phenotypes. We have phenotype, and because we use GO, we have these GO associations with our genes. And so that allows us to kind of triangulate into phenotypes that may be associated with both our chemical exposure and our disease. So this got us thinking more about adverse outcome pathways and whether this information could be used to sort of compute some of these things, and not necessarily saying this is exactly what's happening, but if you wanted to sort of narrow in on possible pathways that would lead from some exposure to an outcome, maybe our data could help with that. And so this is just showing you some of the stats on the data depth we have. And as an example, we've played around with this, and we have a very recent publication, Toxicology, if you're interested in the details, but able to go from a chemical to a number of initiating events. We can sort of take the phenotypes at various levels, so from a subcellular to a cellular to a system-wide level, and then connect those to a disease. And then because we also have this exposure information, you can add that on top of the sort of mechanistic information getting from the exposure to the outcome. And that would be based on epidemiological studies. So I think this combination of the exposure data, again, with the phenotype and mechanistic data could be potentially useful for developing some of these AOPs computationally. And this is just a small snapshot of being able to not only connect these phenotypes with diseases, but getting a lot of mechanistic information in there to help justify those connections. So some of the challenges that we're left with are, you know, and preaching to the choir again, is establishing consensus across these vocabularies. So even among us, and we're probably more connected to this stuff, we're all still not using the same approaches for that. And I don't know what the solution is, you know, because these infrastructure projects take a lot of work. Usually we don't have a lot of money to do it or a lot of people to do it. And so trying to figure out, okay, when do we step back and actually figure out, can we converge on a similar thing, a similar set of vocabularies? Because I think once that happens, all of our resources could be connected a lot more easily. So incentivizing FAIR, building accessible tools. We had a huge bottleneck with just figuring out how to work with ontology development tools, and Chris had to hold our hand through several workshops before we were able to actually get some of our ontologies into, or our structures into a way that we could actually share it with other people if they wanted it. And just cultivating these collaborations. So this has been a really interesting meeting. I know 5 years ago, an ontology talk would be a one-off at a meeting. So it's interesting to have a whole day that's devoted to this. But how we cultivate that into ways that we don't just go home and keep doing what we're doing, and instead, you know, kind of work together in more substantive ways. And I think there's a lot of work being done in that way. But again, I think just from my perspective, we have a small team. There's no extra money. There's no extra bandwidth on staff time. And so I don't know what that answer is to get over that hump. So I'll just end with this. We have a pretty small but very nimble team working on the database, and we're funded by NIEHS and some funding from NC State, and I'd be happy to